This conference will now be recorded.
Okay, folks, so good morning. We are here today on our second class of the semester, and we are going to be going over a few chapters. These beginning chapters of the text and of this course really help to build a foundation upon which you will be learning about the different body systems. But know that really right at this point in time, actually, let's look at something right here. At this point in time, what you're doing is you're trying to understand. I think this is a good image to help you understand why it is that we do start out with what we do as far as with this information here. And that's not going to help seeing it that small. And it's not that clear, but I apologize. I'll have to I'll try and find another image. But look at this here. Here we have the atomic level, okay? And here we have, as far as you're looking at DNA, and we're looking at more of chemical level. So this is really the basics upon which the body is produced and comprised of. So if you then go to here would be the cellular level, tissue, organ, organ systems, and the organism, okay? It's just a bad picture. Let's see if we can get another one here. Okay, this one we can see a little bit better. Make it bigger. Yeah, there we go. That's good, right? So you'll see here, if we go in reverse, the organism is comprised of organ systems. And last week we looked at the different body systems as far as the skeletal system, the muscular system, the nervous system. These are all examples of the organ systems in the body. Those organ systems are comprised of organs. And so these individual organs are comprised of tissues. And in chapter 5, we'll be learning about the tissues and epithelial tissue, muscle. This would be representative of smooth muscle, okay? They're spindle-shaped cells. These tissues are comprised of cells. And here we have this spindle-shaped muscle fiber, smooth muscle. You have no control over it. It's present in the organs of your body as well as in the blood vessels such of your body, okay? And then the cells are comprised of this chemical level, we would say, and comprised of atomic structures, atoms. And we'll look at the atomic structure in a moment. You'll see also complex molecules and such. So do you see how we go from the very foundations and continually build up till we get to the organism? So just giving you an understanding of why. So we're going to be looking at the chemistry of life. We also have cell structure, and chapter 4, cell metabolism. So we'll be looking at these chapters today, and like I said, possibly chapter 5. We'll see how it goes. So let's start off with the PowerPoint here for this chapter as far as looking at the chemistry of life. So cells, tissues, organs composed of chemicals. And so this is the issue that we're going to be looking at as far as you all have heard of, and I'm sure you've probably seen in schools, the periodic table of elements and such. And so let me show you one more thing and then we'll. Yeah, there we go. All right, so here's the periodic table of elements. So you'll recognize, so we have hydrogen right here. So there's carbon, right? Nitrogen, oxygen, right? So these are the elements, and there's gold, there's silver, there's all these elements are present here. Here we have phosphorus, we also have potassium, we have helium. These all elements, right, are really the foundational elements that make up and comprise who we are, the atmosphere that we breathe. I mean, it comprises all of the different structures present within the earth in our bodies. universe, right? These are all, and I'm sure there are elements that we have not been able to identify yet, but these are the ones that we work with right now. So, as we're seeing here, elements, compounds. Compounds are when we have different elements will come together to form some type of structure. Reactions are what take place all around us, as well as what's going on within the human anatomy and such. And so these chemical reactions we'll discuss, and as far as the molecular structure of the different parts of the human body. So the atomic structure, we're looking at protons, neutrons, and electrons. Right? The nucleus contains protons and neutrons, and electrons really are very, very extremely small structures, atomic structures, that are continually in movement, in motion. Okay? So when we talk about the weight of an atom and such, it's really of an element and such, and at the atomic structure there, as far as atomic weight, atomic mass, we're looking at protons and neutrons really comprise the mass of the atom. The electrons, they're so tiny and small. Yes, they're continually moving, but they're not really contributing to the mass of the atom. Elements, compounds, isotopes, this periodic table of elements that I just showed you before, right? So an element, atoms contain the same number of protons and electrons. Okay? And so you need to know that the protons and the electrons, they are the same number as far as how many are present within the element. But the neutrons, they can vary as far as their number. Okay? Two or more elements coming together, that creates a compound. And you can see here that also an isotope, we can have isotopes for hydrogen, we can have isotopes for the different elements, and what can vary is the number of neutrons that are present. Orbital levels, these are the levels upon which the electrons are actually moving, and what degrees of, what's areas of the atom, that the space that the atom is comprised of, where these elements are actually, how many electrons are moving and how many electrons are present, and where they are as far as the levels in the fields where they're moving. It's kind of a interesting thing here, folks. Let me show you as far as you'll see here that depending upon how many electrons are present will depend upon where they're moving outside of the nucleus of the atom. So here you'll see, this is a little bit better. It's a matter of that, here we have just in the first, trying to get a better picture here, folks, to give you an understanding here as far as the levels upon which that they're moving. Here we go. Here we go. That's a little better. All right. So let me make this bigger. All right. So what you're looking at here, folks, is that we have at each of these different colors, okay, this would be where the electrons, and depending upon how many electrons are present, are actually moving outside of the nucleus. So here would be the nucleus, okay, the nucleus of this atom. And then we have, at the first level here, we have 2 electrons that are moving, okay? And this needs to, in order for this atom to be stable, right, it needs to have each of these fields that they have the number of electrons that are present, they need to be full, okay? Now this outer field right here is not full, okay? What you'll see is that we have, at this first level here, we have 2, and this is full. The next one, we have 8, right? So these are present there. Then we go to 14, the next level. And then we go to the next level where we have only 2 present. And so what can happen with these atomic structures is that when the outer shell right here, this shell, is not full, it can actually interact with other atoms and create compounds and such. And I know that's kind of like a little bit out there and such, but just know that this would be then an unstable situation where this atom will be able to interact with another atom, and these electrons can be shared. These shells here can be full as a result of interacting with and bonding with other atoms in the atomic structure. That's about what I can give you there with that. So these orbital areas, this is where the electrons are found. So I just showed you that in that image there, you see in the different levels, right? There are these electrons that are present. Again, the electrons are very, very tiny, very, very small. They do not contribute to the mass of the atom. The energy levels are these groupings of these orbitals, these orbits, right, that these electrons are moving in, and represented as far as, for our purposes, as far as understanding a little bit and learning, concentric circles surrounding the nucleus. The nucleus, again, would contain the protons and the neutrons, right? And yeah, here we go. So take a look at this for a moment here. And what you're seeing here, folks, is that here's a sodium atom, here's a chloride atom, right? And so coming together, what can create this? We can create a salt, sodium chloride, okay, when they come together, okay, when they're bonded together. So there are different types of charges that are present on the protons, the neutrons, the electrons. Now know that the neutron, no charge. Protons, they're positively charged. Electrons are negatively charged. Okay? So know that one atom gains and one atom loses electrons when they form this ionic bond. Okay? So there's a matter of the electrons that each of the atoms, when they come together to bond, sometimes they'll share as a result of these covalent bonds. So the atoms are sharing electrons in their outermost shell. Okay, so that outermost shell here, so you see how we have these shells where these electrons, the circles, these are represent electrons. And here we have, for this sodium ion, we have this sodium ion right here. We'll see that this chlorine or chloride ion, it contains 2, 4, 6, 7 in this shell right here. When we take the sodium ion and bond it with the chlorine atom, then we're going to create a full shell right here, and it's going to be stable, okay, in comparison to then the case where you have only this one atom electron here in the atom, for the sodium atom, and it looks to fill this field. It would be stable if it were to have 8 electrons in its outermost shell. This outermost shell needs 7. Well, chlorine atom does have those 7 that it can work with the sodium atom in order to create a stable environment. So they're both looking for electrons in order to create a stable environment. So in the case of the ionic bonds, one atom gains, one atom loses. In the case of covalent, there's a sharing that takes place of the electrons. In the case of hydrogen bonds, we would say that these bonds are weak bonds, and they'll be able to hold water molecules together. There's a cool experiment. And it's regarding the bonding of these hydrogen bonds, okay, between the different water molecules. Okay, so look at this image here. So what we're looking at here is H2O. You see that there? So 2 hydrogens, 1 oxygen, H2O. Now, in a cup of water, you would have all of these water molecules bonding with each other via hydrogen bonds. Okay? So here's water, here's H2O, H2O, right? They all come together. They are all bonding together via hydrogen bonds. Okay? And these hydrogen bonds are weak bonds, but they still are bonds that will help to keep the water molecules together. Now, I want to show you something here. This is pretty cool. I can't show you the audio, but what I can show you, I can put it on. I can put it on where you can read the there's just a very quick little attention. Okay, here we go. This will show us. Now, I'm going to put this on. Let's see. Closed caption. So you won't be able to hear it, but you'll be able to see the closed caption as they talk. And just watch this for a moment. You'll think you'll find this very cool. And this is as a result of hydrogen bonding, what's taking place, allowing for this to occur. So watch this for a moment, folks. They're gonna. That's pretty cool, right? Now, if you remove it, because he has a screen on his. So look at what he's going to show you. Those hydrogen bonds of the water molecules. All right, so that's just showing you a little bit regarding the bonding that is taking place between those water molecules. And you can do that at home. It's kind of cool, actually, to be able to do that. So come back here. So you see here, as far as there are electron donors, acceptors, carriers. So this is the deal as far as with the electrons are concerned. The electrons are really what allow, whether we have them or don't, whether atoms can come together and share them, give them, whatever may be going on as far as this is what's contributing to the creation of these compounds and such that are present within nature and such. Like I said here, showing you sodium chloride, as far as showing you that salt can be produced and other different types of compounds and molecules, right? But just know that it's the electrons that are being shared or being given to, right? Atoms gaining or atoms are losing electrons. It's the electrons that allow for the ability for bonding to take place between the different atoms. As simple as that. Here you're seeing as far as two hydrogen atoms, okay? And so they have one electron in their field, okay? And this shell right here, there's only one electron for hydrogen. Here's one electron for hydrogen. When they come together, folks, then they will help to fulfill that requirement for two electrons that need to be in that first shell, okay? That first shell, see here that now they're sharing those electrons, okay? So this is different than a hydrogen bond. This is two hydrogen atoms coming together, okay? So when we looked at here, as far as, again, looking at that we have an oxygen and two hydrogens, okay? There's a bond, there are bonding that takes place here. But look at here, folks. These bonds that are taking place between the water molecules. So here's two hydrogens and an oxygen create a water molecule, okay? These water molecules will then bond with each other via these hydrogen bonds, okay? And so that's what's taking place as far as the different types of bonding that's taking place within these structures here in water. Look at this image here, and it's showing you that here again, water, two hydrogen, one oxygen, and there are covalent bonds. Remember I said to you, showed you this image right here. Covalent bonds, atoms are sharing electrons in a covalent bond. Well, here there's sharing that's taking place of these bonds right here. Electrons are being shared between oxygen and hydrogen, hydrogen and oxygen. That's a water molecule. When these water molecules all bond together, they bond via hydrogen bonds. Okay, so it's a little weird here. So you're seeing here in this image, I have a water molecule, covalent bonds, there's sharing of electrons, covalent bonds, there's sharing of electrons, and here we have a hydrogen bond that is bonding two water molecules. Okay, let's come here. So when we're looking at water in the human body, you are comprised of quite a high percentage of water, and all of the cells of your body have some type of water volume to them. As far as we're going to be looking at the next chapter, the cells have what's called the cytosol. It's part of the inner workings of the cell. When we look at it, I'll talk about that in more detail. But just know that water is in all of the cells of your body. It's also present within the blood, right? The blood, the plasma, the liquid portion of the blood, comprised of water. Now there are other chemicals, there are other ions, there are other parts there are other things that are present within the blood as far as carrying of oxygen, CO2, carbon dioxide, in addition to the blood cells and the plasma, the liquid portion of the blood. But just know that water is extremely important, okay? Most abundant substance in cells, universal solvent. We're going to be talking about solvents and solutes in a little bit as far as the movement of material that will take place in the human body, as far as whether we're talking about diffusion, we're talking about osmosis. This is how nutrients enter into cells. This is how oxygen and CO2 are carried in the blood and brought to the cells of the tissues of the body. Very important as far as movement of material. We'll look at that also today, that transport of materials. Really, as far as in the plasma is concerned, very important. Absorption and reducing heat. So what will occur, folks, is that we can sweat in order to cool the body down. very important as far as that's concerned, as far as temperature regulation. We'll look at that and throughout this semester. And also protection of body structures. Really know that cerebral spinal fluid, this is a very thin, watery substance that is present in the brain and spinal cord, as far as present within and present outside of, and as acting as a protective buffer for the nervous system. Carbon dioxide is a waste product of cellular respiration. You won't really need to know a whole lot about regarding what's taking place with that, but know that the body needs to produce ATP. It's called the energy currency of the body, of the cells and of the body itself. Really, ATP is needed for many functions within the body, and actually movement of materials against what's called a concentration gradient, against the way that they should naturally just go without requiring any energy. We need ATP. So cellular respiration, very important to produce this structure called ATP. Well, as a result of this production of ATP in the cellular respiration, CO2 is a waste product that's produced. It's used in photosynthesis to produce usable energy sources. So in the case of talking about plant life and such, absolutely important. Must be removed quickly from cells. And really, how do we remove it? But we're actually then, as we inhale the air and surrounding air in the atmosphere around us, which I have to tell you, right, does contain oxygen, absolutely, but it's a smaller percentage of oxygen than you would think. Primarily, it's nitrogen gas, but we're taking in oxygen, and then the body will do what it needs to do as far as taking it to the cells and tissues of the body, and then it'll remove CO2 and we'll exhale CO2. So carbon molecules come from carbon dioxide gas, and we are carbon-based life forms, folks. So carbon is very important as far as in the structure of proteins, carbohydrates, lipids, and fat. Lipids are fats. Okay. Molecular oxygen, so formed from covalent bond of two oxygen atoms. So here you're seeing, so when we think of oxygen, we would say it's O2, okay? Two oxygen atoms, required by all organisms that breathe air, right? We know that, we get that. Without that, you have a problem. Have you ever been in a pool or in the ocean or in a lake or wherever, and you go underwater, you hold your breath for a period of time, right? Your body needs oxygen. Now, know that it's not really the desire for you to need oxygen that you would come up out of the water and like, no, it's not that. It's really the buildup of CO2 present within your body that's triggering you to say, hey, I need to get up out of the water and breathe air, okay? But it's really, you need to be able to exhale the CO2 in order then to take in the oxygen and such. See here, necessary to convert food into ATP. That ATP is a very important structure. Like I said, it's the energy that's required for the body to do its functions. For you, so I'll talk to you about something interesting for ATP. So a cadaver is a dead body, right? And we'll use those in our graduate schools, and I spent a year, and Dr. Massimo, I'm sure also spent a good portion of her undergraduate training, but really like first year training in graduate school as far as looking at and studying cadavers. Now, know that the cadavers have been preserved and such with different chemicals and such, and so it's a little bit different looking at a cadaver and then looking at a recently deceased body. Right, that's not been preserved. So a recently deceased body that's not been preserved, after a period of time, the musculature can become stiff. Okay, and have you ever heard this term? It's not a respectful term, but they'll refer to a dead body as a stiff. Okay, because the musculature has contracted and it's not relaxing. The reason that's taking place is because your body is not producing ATP. And because it's not producing ATP, the muscles have contracted, but they can't relax. Okay? ATP is necessary for the actual separation of the different proteins present within the muscle that allow for it to contract, right? So you contract your muscle, you can feel that it gets hard. contracted, but you can also then you can just stop that and you can allow it to relax and then it becomes soft. Well, in the case of a dead body, there's no longer any type of metabolic processes going on. So as a result, there's just going to be a matter of the ATP is not produced. So there'll be ability for contraction. It'll begin because there's still going to be chemicals present that will allow for this to occur. But the chemicals that need to be produced in order for the muscles to relax are not present. So hence, we do need ATP for that, in addition to many other processes within the body. And I mentioned that the level in the atmosphere, right? It's a smaller percentage than you would think. So 20%, that's oxygen. Really, like I said, nitrogen is a very high percentage of the atmospheric air that we breathe in. Ammonia. Now, when you're thinking of the human body as far as producing waste, so we'll produce liquid waste and we'll produce solid waste. And as a result of protein metabolism, the breaking down of proteins, right? The catabolic activity, we would say, that's tearing down proteins and then being able to then synthesize new proteins with those building blocks. Folks, the bottom line is amino acid breakdown. It's the protein metabolism. Proteins are made up of, comprised of amino acids. Ammonia is produced. So you see here, amino acids, building blocks of proteins. Amino acids contain nitrogen. And so what will occur is that this ammonia is a byproduct of this, and it needs to be removed. Mineral salts, okay, composed of small ions. So ions are specific structures that have a charge. So sodium and potassium are very common ions present within the body. They're both positively charged, and they're both very important for your nervous system to be able to send messages throughout the body. And they're also important for the muscular system to be able to contract. Very important. So sodium potassium ions are extremely important, and their concentrations, their levels of those sodium potassium ions is a delicate balance, and it needs to be maintained. Recall we talked about homeostasis last chapter. So really, folks, calcium, phosphate, sodium, potassium, chloride ions, these are all very important in order for these ions to be present within the body to perform the functions that they need to perform. And if they're not present, and if they're present at different levels or lower levels or too high levels, illness can occur and improper function can take place and lead to unhealthy environment and lead to a lack of homeostasis, really, where a stable environment within the body, no matter what goes on outside of the body. Okay, so these are all very important. And what you'll find also is when we're talking about the skeletal system, that calcium is stored. So today was payday for me, and so my check was deposited into my account, and so the bank holds my money, right? And so when we need the money, we take it out, do what we need to do. When the body needs calcium, because the body will need calcium for muscle contraction as well as for the nervous system, and it's a very important mineral salt, an ion. Where does it go to? Well, yes, nutrition, taking in calcium. So I had a yogurt this morning, and so that will contain some level of calcium. But really, when it needs calcium on a regular basis, whether I'm eating or not, calcium bank would be the skeletal system. Your bones contain levels of calcium that through hormonal action, we can actually remove some of that calcium from the bank of the skeletal system and use it for the functions of the body. So right now, what we're going to look at is the different types of organic molecules that are present within the human body and in other creatures also. So carbohydrates, lipids, which are fats, proteins, nucleic acid. And nucleic acids, this is the genetic material inside of all the cells, okay? And this really, DNA, RNA, is very important for the ability to produce proteins, okay? Now, understand that the human body, we take in animal and plant carbohydrates, proteins, fats, nucleic acids, which are present within the cells, right? The nucleus of the cells. And we take them in, and then we digest them, we break them down and convert them to be able to produce human carbohydrates, proteins, fats, nucleic acids. So it's pretty cool how the human body is able to take this material, this raw material, and again, if we're a vegetarian, then we wouldn't take animal products, but we'd be taking in the plant products, and again, taking those plant carbohydrates, lipids, fats, the proteins, the nucleic acids, and converting them to human organic molecules and such. So know that carbon, I said that we are all carbon-based life forms. Carbon is extremely important and a part of a major component of all of these organic compounds here. Okay? You'll see that there are functioning for carbohydrates, energy storage, cell structure. They help to maintain the different structures present of the cell itself, and we'll look at the cell in the next chapter, okay? But just know that all of these have a major component in the structure of the human body, right? They're the really lipids, very important for the plasma membrane, the actual covering of the cell. And so it's really important that we take in also, as far as nutrition is concerned, all of these different, so not trying to just limit, oh, I'm only going to eat this type of food or this kind of type of food. There needs to be a balance of the different foods that we take in. Know that we know that lipids, fats are insoluble in water. You've seen salad dressing, right? And know that it's comprised of water and different spices, as well as oil and vinegar, right? And so the vinegar will contain water, and the oil, right? You'll notice you have to like shake it to get it to kind of mix a little bit there in order for you to then put it on your salad. But it really does not stay in solution, right? The vinegar portion with the different, spices and such will separate from the oil. Insoluble, we would say it's really hydrophobic is a good word to put fats. Water fearing. 95% of the fats in the body are triglycerols. Saturated and unsaturated fats. When you think of a saturated fat, think of butter. Think of lard. That's a saturated fat. Unsaturated fats, think of oils. So olive oil, right? Canola, vegetable oil. Those are unsaturated fats. Energy, insulation, protection. As far as the body is concerned, so know that the eyes and also the kidneys have a fat cushion, a lipid cushion, an adipose tissue, more appropriate term, adipose tissue. This is fat tissue. Adipocytes are fat cells, and fat is present in the eyeball, right, posterior to it, in order to cushion the eyeball. Also, the kidneys need that fat cushion in order to be protective area there. And if a patient were to go on like a starvation diet or become anorexic and lose a lot of weight, you'll notice that the eyes will kind of sink in a bit. And what can also occur with the kidneys is that the kidneys can actually drop, and there can be a kinking of, like you have a cord and it kinks, like a tube that kinks. Well, imagine the tube that contains urine that takes it from the kidneys to the urinary bladder. Imagine if that were to kink. Then what can happen is that there can be a buildup of fluid in the kidneys as a result of not enough urine passing to the urinary bladder. That can be a problem. This is just as a result of a lack of that fat cushion for kidneys. So really, insulation, protection, providing a form of energy as far as nutrients. When we say we're out in the desert and we haven't eaten for a couple of days, we'll know that, right? What'll happen is that your fat stores will be used, the adipose tissue will be used for nutrition before, then as time goes on, if the body uses up all the fat stores, it'll start then breaking down proteins and such at the body. Now the proteins, really, folks, the proteins are everywhere throughout your body, very important. Again, these are all carbon-based. So all of these structures are carbon-based, okay? And you'll see here that these proteins are made up of amino acids, okay? Let me take a look here for a moment. I'll show you. Right here. I'm going to make this bigger. I'll enhance the image. And let's make it bigger. And what I would like you to see is that, let's see here, okay. So here are examples of amino acids that you might have heard of. Arginine, lysine, glycine, proline, threonine, right? You might have heard of these. Tryptophan you've heard of. Tryptophan you've heard of as far as for when we talk about during Thanksgiving time and you're eating turkey, and then as your body is breaking down the turkey, tryptophan can lead to a kind of a calming sensation in the body and such. These are all examples of amino acids, and proteins are comprised of these amino acids. Okay. Proteins actually act also as enzymes, and these enzymes are very important because they are able to enable, facilitate, maintain the different chemical reactions that occur. They're a catalyst. They really help a chemical reaction to occur within the body at a lower level of energy is required because of the enzymes. And again, these enzymes are protein-based. Okay? They have a very unique structure and provide energy. Really, proteins are very complex. They can be very complex. Let me show you here. Okay, here we go. So as you're looking at this image here, you'll see that there is a primary structure. So this is a chain of amino acids comprised of this basic protein structure. We can have this helix or this pleated sheet type structure. Then this tertiary structure can be a little bit more folding that takes place. And this quaternary structure, the more complex of the protein structures, where we have a lot of folding taking place and multiple, these structures are all present together. So proteins really can be very complex structures depending upon their function and the need for what they need to do. Proteins also can be, let's see. Here we go. So proteins also can be disease causing when they're not, when they're folding. Here, when they have, here we go. Look at this first here. Have you ever heard the term prion? You might not have heard the term prion. It's a disease causing protein, a pathogenic protein. Now you'll see here that there's folding that's taking place in this normal protein present within the brain. The disease-causing form of the prion protein, it's different. It has different structure to it as far as how it is and the folding that's taking place. And just as a result of this difference from this to this, right? This is normal, abnormal. This can lead to catastrophic illnesses. You've probably heard of mad cow disease, right? So mad cow disease would be this, an example of mad cow disease in humans would be this prion type diseases. And there are different types of these diseases, CJD, V variant CJD, present within humans that as a result of an interaction with these prions that can create all kinds of a catastrophic end. Okay, it can lead to death. Scrapie, you'll see here this bovine spongiform encephalopathy, that mad cow disease, chronic wasting disease. In the case of deer and other types of those types of like elk and such also can have chronic wasting disease. These affect what takes place within the brain. I don't have another image here. Yeah, here we go. So what's taking place, right? So Creutzfeldt-Jakob disease. So this is that prion, these pathogenic proteins that can be present within the brain. Creutzfeldt-Jakob disease is caused by abnormal proteins called prions. They're not killed by standard methods for sterilizing surgical equipment. As prions build up in cells, the brain slowly shrinks and the tissue fills with holes. Okay? So you see these spaces in the tissue. The brain slowly shrinks and the tissue fills with holes until it resembles a sponge. And so this is like this term here that we saw over here, spongiform encephalopathy. This is the change that's taking place within the brain of the cow as a result of these pathogenic proteins. Lose the ability to think, to move properly, and to suffer from memory loss. Always fatal, usually within 1 year of onset of illness. It can be longer, and it can be shorter. So it's really so understand that when a protein is not folded properly and not in its correct normal form, it can lead to devastating circumstances for the human, for the animals. It's horrible. And here we're just showing you, as far as I just showed you an image before regarding the structures of proteins, whether it's very simple to more complex at the quaternary level. Nucleic acids, folks, this is DNA, RNA. DNA and RNA are very important because they really, the nucleus of the cell, we'll see in next chapter, contains the genetic material of the cell, the DNA, and RNA is present in order to allow for protein synthesis to occur. And really, RNA is a copy of DNA present within the nucleus that will in turn perform this protein synthesis, which is vital for the life of the cell as well as the organism. So there's a double helix for the DNA and a single for the RNA. So we have here okay. So just look at this for a moment here, and you just see that there are, so DNA, right? Here we have 2, and here in RNA, there's only 1, right? So we only have this one single structure, as far as DNA has this double structure here, okay? And I'll mention to you regarding these bases. So these bases, depending upon how they're present on the chain here, okay, will then be first transcribed to RNA. There'll be a match to these. We'll separate. It's called transcription and translation. Transcription is where we're going to take one of these chain and match it with an RNA chain, and the bases, these structures right here, will match and create the RNA that will then leave the nucleus of the cell and go out into the cytoplasm and be involved in producing proteins as a result of this chain of these bases, which will match with, which will really create and link with amino acids that will form proteins that the body needs. So again, DNA has this double chain, RNA has this single chain. They're made up of these chains of these nucleotides, which contain a nitrogen base, sugar, and phosphate group. And that nitrogen base, that's what I was showing you there as far as those purines and those pyrimidines. That's that image right here. Right here, as far as looking at this. I'm gonna take this image. Just make it bigger, just show you. There we go. Okay. And I can share this with you folks. You can look this up also, but you'll see here that these are the nitrogenous bases. Okay? Nitrogenous bases, and what will occur is that you'll see how there's a pairing that takes place. So the C and the G go pair together, the A and the T pair together. See that here on DNA? So here the T and the A and the T, C and the G, right? They pair. Now, when I separate the DNA into one strand, this strand then will look to create an RNA strand that will pair with what we have here on one of these strands. Now, know that in the case of RNA, it only contains uracil, not thymine. So any A or T from here that comes over and tries to form a base pair will not be able to form a base pair with the T because only U is here. So you'll see here that it's a little bit different as far as how we're creating this strand here that will go and perform out in the cytoplasm, out in the area that's outside of the nucleus of the cell. It'll actually work to perform and create a protein. Let's see. In this class, folks, you are looking at this information. It's not a chemistry class, and it's not. We're trying to give you all of this information in a short period of time throughout the semester, and know that like the level that you need to know this, focus on what you have here as far as in the PowerPoint. Trying to give it to you in a situation where there's a lot of complexity to what we're studying today, and yet you are not required to know it at such a very complex level, but just at a very basic level. And so if you focus on what you have here in your PowerPoint, as far as studying purposes are concerned, you'll be fine. This ATP, this adenosine triphosphate, right? Very important as far as for the, like I said, the energy currency of the body. Any type of energy that's required for the body to do its processes, ATP is what's being used. Okay. Okay. So movement in and out of the cell. Okay. The plasma membrane is the actual outer covering of a cell. Okay. Meaning selectively permeable means that it's going to allow for certain substances to enter in and other substances not. Okay. Water can enter and exit with ease. Okay. Fats can enter through the plasma membrane very easily, but other water-based molecules and such cannot. Okay? And ions need specific types of channels. They need little gates. They need openings to allow for them to pass through the plasma membrane and enter into the cell, okay? Or exit the cell. You'll see here this image. You didn't have this image. I posted it. I added it to this PowerPoint here. Active and passive transport. So active transport, we need energy. We need ATP in order to allow for material to move back and forth. But there's a reason why we need ATP. Let me bring you over here. On this image, you're seeing that extracellular fluid. This is outside of the cell. Cytoplasm, intracellular. This is inside of the cell. Well, recall that I said to you earlier that sodium ions, potassium ions, they're very important for the function of the nervous system, of the muscular system. They're very important because what will happen is that as a result of the different concentrations of sodium ions and potassium ions and the movement back and forth across the plasma membrane, it will create a change within the plasma membrane, which will allow for things to happen. Let's put it this way at this point in time. Now know that passively, no energy required. Sodium is higher concentration. It's bigger here. You see how the lettering is bigger? It's a higher concentration here outside of the cell than here inside of the cell. Okay. Potassium is higher concentration here and lower concentration outside of the cell. So sodium ions will move with no energy required from outside to in. If there's this channel, this leak channel, it's an open channel, it's protein, and it allow for movement from these sodium ions to go from high concentration to low concentration. That's called diffusion. Potassium ions are higher concentration inside of the cell than outside. And so they'll move from high to low. But what am I seeing here though? This says active. So moving against this concentration gradient, right? From low to high, from low to high, that requires energy, and that's the ATP is required. So if we had one of these over here for potassium, for K, it would be moving from high concentration to low concentration passively, because concentration gradient, it goes from high to low, high to low. See this lower, smaller K? That's passive. But this is going against the concentration gradient from low to high, and that, folks, is as a result of requires ATP. And this is all involved in really changing what takes place as far as the resting membrane potential of this plasma membrane. Don't worry about that right at the moment. Let's just move on here. But active and passive, if it's an active process, we need ATP. If it's a passive process, no energy is required. So moving of molecules from area of high concentration to low concentration, this is diffusion. Okay? Temperature can increase the rate of diffusion. Okay? Oxygen and CO2 movement is as a result of diffusion in the body. So high concentration of oxygen in the lungs, low concentration in the blood, and then the blood will receive that oxygen. That's diffusion, that's movement. CO2 coming from the blood and then into the lungs, diffusion. Here I provided this also for you to show you that. So these are solutes, the blue dots, and then the gray would be the solvent, okay? Recall we said that water many times is a solvent. So here's high concentration, low concentration. So if we're going to move from high to low, where are these solutes going to move? They're going to move across the plasma membrane into the inside of the cell. And so here we have equilibrium. It's equal concentration here and here. Now, if I were to move the liquid portion, the gray area here, outside of these little blue solute pieces, that would be osmosis, the movement of water, the solvent, through the semipermeable membrane from high concentration to low. So again, it's diffusion of water. Osmosis, the diffusion of water. You'll see here where it talks about tonicity, isotonic, hypotonic, and hypertonic. So let me talk to you about, I went forward for a moment here and showing you hypertonic. In a hypertonic situation, this is where we have the salt concentration is very high outside of the cell in the plasma of the blood. This is as a result of a situation where a patient is drinking salt water. So if you are, a person is drinking salt water. So say if you're stranded on a desert island or you're in a boat, right? And you have no fresh water and you don't think to take care of removing the salt from your water, and you just drink salt water, well, one, it'll taste nasty, and two, it'll slowly kill you. And the reason being is that because it'll create a very salty environment within your blood, to the point where then the water that's inside of your cells, your blood cells, your red blood cells, will come out of the cell and enter into the water because of osmosis. And as a result, it's going to kill the red blood cells. Okay? So you'll see here salt concentration higher outside the cell than inside. And that's because you've drank this salt water, and eventually it'll kill all of your red blood cells, which will lead to the inability to move oxygen to the cells and tissues of the body. And this is quite bad. That'll lead to death. Isotonic, when you think of saline solution, that is a very slightly salty solution, and that would be an isotonic type of solution. Salt concentration is the same outside and inside of the cell, and so water can move in and out of the blood cell, and it's not going to have any detrimental effect. It's not going to damage the cell whatsoever. In the case of hypotonic, the salt concentration inside of the cell is higher. So if you were to drink just tap water, spring water, and if you were to drink a lot of it in a very short period of time, it could lead to a hypotonic environment in your blood, which could lead to your blood cells actually filling up with water to the point where they would burst. So hypotonic, hypertonic, not a healthy environment for your cells. Isotonic solution is the way it should be within your body. This is a better example. I like this. So see here, as far as the pink represents the water, okay? The little balls here represent the solute. Now the solute cannot pass through this semi-permeable membrane, but the solvent, the water, can. So watch this here. So we have high concentration of water, low concentration of water. It's going to move from high concentration to low until we get to equilibrium, where it's the same concentration of between water and these particulate matter, the solute. So solvent, pink, solute, the little dots there, same concentration on both sides of this semi-permeable membrane. And again, this term active transport, you require energy. So that's the ATP that you'll require in order to do this. And so it's an active form of transport, whereas diffusion, osmosis, we're looking at a passive transport. Now, pH, this is very important as far as understanding that the level of hydrogen ions present within some type of liquid. Let me show you this here. So this, look at this. I like this image better. So you're seeing here, as far as 7 would be neutral. So water is considered neutral. Higher pHs, this would be alkaline or basic. Okay, so your blood is 7.4. Coffee is slightly acidic. A tomato is acidic. Vinegar is a weak acid. Right. So we get to like hydrochloric acid, battery acid, very, very low pH. Here we would have high concentration of hydrogen ions. Here, low concentration of hydrogen ions. So you'll see here an acid base, a buffer will help to maintain pH levels so that they're not, so that buffers present within the body, in particular in the digestive system, will help to increase the pH so the material that's being digested and broken down in the stomach is very acidic. The stomach is a very acidic environment in order to really destroy microorganisms. Buffers can be released by the pancreas into the small intestines in order to raise the pH so it's not so acidic as it's entering into the digestive system as far as the small and large intestine. Okay. All right, folks, very good. So that's our chapter 2 as far as the chemistry of life. Okay, how are we doing? I want to continually move on and such, but how about if we take like a couple minute break? Need to just take like a 3-minute break or so. Restroom, get a drink, whatever you need to do, and then we'll start in 3 minutes. How does that sound? And what I'll
That sounds good.
sound good? Okay, very good. So 3 minutes. Okay, we'll come back in a couple minutes and we'll continue on. Very good. Okay, folks, we're going to come back to now our PowerPoints, continuing to record. I'm going to move on. Are there any questions at this point in time? I know that was a lot of material, and I know that chemistry, if you're not a chemistry major, it can be dry, and I apologize, and I try to bring a little bit of interesting points regarding the different disease and such and how it can pertain to the body, but yeah, chemistry can be a little challenging at times. Yes, Lauren, go ahead.
I don't have anything to say. I was just listening. I took chemistry once before and I failed it.
Yeah, it's it's not for everybody. I had to take many different types of chemistries. It wasn't my favorite subject matter, to be honest, but it's okay.
Yeah.
You do what you got to do.
Yeah.
Absolutely. All right, folks. So I'm going to hide everyone then, and then I'll continually finish up as far as chapter 3 is concerned. Go into chapter 3. We're going to go until about 10:30 there, folks. Okay, so we should be able to get through these slides in a pretty good clip here. So you'll see here, as far as the cell is concerned, as we're talking about the cell, it's the basic unit of biological organization. And we'll see that there is the cytoplasm that's present, this protoplasm. It's the inner portion of the cell. We have the cell membrane, and within this protoplasm, the cytoplasm, we contain the organelles. And so we're going to be talking about the structure of the eukaryotic organisms. These are cells that have organelles. You'll see that the prokaryotic cell really has no organelles, no membrane-bound organelles. Okay, so you're going to see the membranes that are present for the different tiny organs, right? So the organs of the body, right, are analogous to the organelles of the structures present within the cell, and we're going to talk about that today. Look at that. Back in the 1800s, Schleiden and Swan came up with the cell theory and such, and so in the study of the cell, and so now we are going to be able to look at and focus on and really see cells arise from other cells. We're going to be talking about in chapter 4 mitotic division, and meiosis, and this is cell division, taking one parent cell and creating two identical daughter cells. Okay? So here we have an image of the cell, and you're looking at the nucleus and the nucleolus, and these are regions that contain the DNA and then RNA produced in the nucleolus of the nucleus of the cell. We have these different folded membranes that will be present in order to produce proteins, produce carbohydrates and lipids, and then package them for either use inside of the cell or out or on the plasma membrane, it's the plasma membrane, or outside of the cell. Okay? You'll see here that we have structures called mitochondria, and these are a membrane-bound structure. You'll see here these are also membrane-bound structures. Okay, that's what creates a eukaryotic cell. That's what makes it different from, and this is also the nucleus. This is its nuclear membrane, and so it will allow for then the structures to stay within side of these membranes present within the plasma membrane, the cell membrane of the actual cell itself. Okay? We'll see that all these structures have specific functions and such, and so we're going to go through that, and you'll see that that's going to go by pretty quickly. I am going to require of you all today for next week, okay, and then you'll have this assignment with Dr. Massimo. It'll be in coursework, but I'll put it in coursework for us today. As far as you're going to watch a video of the active and passive transport, and you're also going to define the different organelles present within the cell. You're going to define these terms. So I'm going to have these terms for you that you will create a Word document and you'll define them. So there's going to be 2 assignments due for next week. Okay, and let me show you here as far as, here we go. So I'm going to share this link with you all. Okay, I'll share this link with you all. And this is the video that I would like you to watch. It's on active and passive transport. And you're going to just give me a brief summary of the video. Okay, it's a 6-minute video, but it's a very good video, and it'll help you to understand a little bit more regarding active and passive transport, diffusion, osmosis. Okay, so that'll be one of your assignments, as well as, like I said, creating this document, Word document, that will have the definitions for the organelles of the cell. Okay, so just as a review type of a process here for you. So you'll see here that this cell membrane, also known as the plasma membrane, is a fat. See, it says here a phospholipid. It's a fat, okay? And it has proteins present within. I showed you that image of the active and passive transport in the other PowerPoint, and that showed you that there are proteins present that allow for, these proteins will allow for movement of material from outside of the cell into the cell, or inside of the cell to move outside of the cell. So the blue represents proteins. Membrane transport, so selectively permeable. So we talked about these, and so know that that term, the fluid mosaic model, that's what this represents. Okay, it can be, we can damage this, and it can be repaired. Okay, the plasma membrane. Okay, and again, know that material to move in and out of the cell requires these proteins, except for water. Water can move through, and fats can actually penetrate through, because this is a fat. You see these little dots and these little strings here? Well, this represents a phospholipid. This represents a phospholipid. This little dot and these strings. So those structures are a phospholipid bilayer, meaning that there's 2 layers of these fats present to create this plasma membrane, this covering of the cell. So look at this here. You're not seeing it here, but this is the plasma membrane in kind of orange yellow. That's the plasma membrane that contains all the guts of the cell. All of this, the cytoplasm, the protoplasm, the organelles, the nucleus, that's all present within this cell and held together via this plasma membrane. And, yeah. So I'm going to move right to the nucleus and such. I don't think that you're going to really need to have an understanding or really go through this as far as solution and colloid, but just know this, folks, that the plasma membrane is very specific as far as what it allows to pass through and what it doesn't. And what it doesn't, then it will need a channel, one of these protein channels, in order to penetrate through. Or what will happen is that it'll interact with a specific receptor outside of the cell that's in this area, connected to the plasma membrane, that will then cause change inside of the cell. And that's called a G protein mechanism. You're not going to need to know that, but just there are other methods of making change within inside of the cell. The control center of the cell. So the nucleus contains really the genetic material. So the genetic material inside of the nucleus allows for the cell to produce proteins in particular. And this nucleolus, that's the inner part of the nucleus that contains these ribosomes. And the ribosomes are involved in protein, ribosome formation, protein production, protein synthesis. I'll show you that in a moment here. Mitochondria are the powerhouses of the cell. They produce ATP. Mitochondria produce ATP, that energy for the cells. Okay? They have these folded membranes. You're going to see in the cell that it's very unique in its structure, and that there are a lot of these folded membranes. Okay? Folded membranes. That's just how the cell does what it does, and how it allows for the production of that ATP in particular, but also for proteins, carbohydrates, and fats. But the mitochondria, only ATP is being produced. And the energy requirements of the cell determine how many of these folds are present and how much ATP needs to be produced. And if the cell needs more ATP being produced, we'll also produce more mitochondria. So there's, in addition to the nucleus of the cell containing DNA, mitochondrial DNA is present within the mitochondria to reproduce more mitochondria, to synthesize, to create more mitochondria. Lysosomes work to act as the digestive portion of the cell, and they'll contain enzymes. Enzymes are proteins, and the enzymes will contribute to the breaking down of material that's brought into the cell, or the actual, if there are organelles present within the cell that are like need repair or that they're old and weak, then it'll actually contribute to the breakdown of those old parts of the cell or the actual cell itself. The endoplasmic reticulum, this structure is a folded type membrane that will either contain ribosomes and it'll produce proteins, or it'll not contain ribosomes and produce lipids and carbohydrates and such. So this rough ER, smooth ER, protein synthesis, smooth ER, really fat transport and lipid synthesis. Look here for a moment again. As we're looking at this image here, so you'll see that ribosomes are these little dots, and they can be floating in the cytoplasm, present in the cytoplasm, as well as connected to this endoplasmic reticulum. Okay, now this would be smooth ER. This would be rough ER. See how it's got the little dots on it? So no dots, smooth ER, dots, those are the ribosomes, those would be then rough ER. Okay? And the Golgi apparatus will take these substances that are produced by the rough ER and the smooth ER and package them for either transport inside of the cell, transport to the plasma membrane to be used as far as the plasma membrane repair or protein channels for the plasma membrane, or to actually exit the cell. So you see here, these are going to then exit the cell and be used somewhere else in the tissue of the body. So that Golgi apparatus will package what's being produced by the endoplasmic reticulum, whether it's smooth or rough ER. Okay? Carbohydrate synthesis also taking place in the Golgi apparatus. So it's proteins via the rough ER, the smooth ER will synthesize the fats, and the Golgi will synthesize the carbohydrates. Okay? And the Golgi will package all of that material and either package it for movement inside of the cell or the plasma membrane or outside of the cell. And again, the ribosomes, these are not membrane-bound organelles. Right? A ribosome is not. There's no membrane covering it, but it is the site for protein synthesis. And it'll work with the RNA of the cell in order to produce proteins. So proteins function as for the structure of the cell, for enzymes, right? Enzymes are catalysts, and enzymes will facilitate reactions inside of the cell, right? Or outside of the cell. And the immune response, there are immune type proteins that are very important for immune function, and particularly for the whether it's specific or non-specific immune function. Right? Your immunoglobulins, which are the antibodies that are present within your body that are going to provide a specific response to specific types of antigens present that could make us sick, proteins. So proteins play a vital force, a vital role in the function of the human body. Know that DNA codes for a particular protein, and it's like a recipe for a protein, and it's found in the nucleus. These terms, transcription and translation, this is what I was mentioning to you in the when we were talking in the other class, other chapter regarding the production of proteins. And transcribing is where we have the RNA will copy the one single strand of the DNA code and then exit the nucleus via these pores in the nuclear membrane. Oh yeah, here we go. So this is the nucleus. Here's the nucleolus. And what will happen is that there's little holes. See these holes? These holes will allow for the RNA to exit the nucleus and then interact with a ribosome in order to then create a chain of these amino acids. So the amino acids are present within the cytoplasm. This RNA, these two different types of RNA, transfer and messenger RNA, will interact with these base pairs and then create an area for these amino acids to come to the transfer RNA and create this chain of amino acids, which is a simple protein. Okay, so again, amino acids are proteins are made up of, comprised of amino acids. Proteins are comprised of these amino acids. So each one of these colors represents a different type of amino acid. So again, transcription takes place here. We're transcribing information from DNA to RNA. We're translating the message from these, using these two different types of RNA to produce this chain of amino acids. So transcription takes place in the nucleus, translation takes place in the cytoplasm, outside of the nucleus, but still within the cell. Centrioles, and these are protein type structures that allow for and contribute to the actual structure of the cell and allow for also the ability when the cell is involved in mitosis. So again, taking one parent cell and creating two identical daughter cells, they allow for then movement of the genetic material to separate areas in order to allow for the creation of two identical cells with the identical DNA. Cilia, flagella, these are important for movement of material across the cell surface, and the flagella important for, it's the tail for the sperm, and now to allow for the sperm to move outside, once the sperm is ejaculated outside of the male penis into the birth canal of the female, then the sperm will then move via the flagella. So that's the only cell within the human body that actually has a tail, a flagella, flagellum. Cilia in the respiratory system, in the digestive system, in the reproductive system, cilia are present. Let me just show you an image of cilia. Right, so cilia and microvilli. This is a good image here. And what you're seeing here is that cilia are on the, see how it's on the surface of the cell? Here's the cell, that's the nucleus of the cell. Here's the cilia. Here are these microvilli. Microvilli allow for an increase in surface area of the cell. They allow for absorption. So in the digestive system, that would be important, to absorb nutrients. In the case of cilia, there's no absorption taking place. They're moving material along the surface of the cell. And here we'd see the cilia. And they can allow for, say, in the case of the respiratory system, they would have mucus present, and this mucus would then connect to and attract particulate matter that you breathe in for the air that you're breathing in, and allow for that particulate matter to get stuck to the mucus and then taken to the digestive system instead of you inhaling it and it enter into your lungs and creating a problem for you. Cilia, very important, okay? Very important as far as their role in the respiratory system and the movement. So it's not moving the cell, it's moving material that has the mucus present along the surface of the cell into the digestive tract. So you can look up, and I'm not sure if you'll be able to access this here as far as the animation, but you can look up on YouTube regarding cell animation and looking at, it'll describe the different functions of different structures present within the cell. Lastly here, you're going to see as far as just giving you some information regarding plant cells and such. And so again, this is we're in like human anatomy class, but just to give you an idea that, yeah, plant cells have some different structures present that will allow for the ability to take CO2 and light and from the sunlight and such, and create chemical processes that will release oxygen into the air and really be very helpful as far as we living together with the plants within our world in order to maintain our atmosphere, and very important as far as also for nutrition and such too. Yeah, so you'll see here, so that the structure that's present within the plant cells, cell wall, this is unique to a cell, to the cell structure. The plant cell will have a plasma membrane as well as a cell wall. Okay, animal cells, right, do not have the cell wall. And lastly here, and this is just an extension of the chapter here, chapter 4, just taking a few slides and just going over with you regarding mitotic division and such. So let's take a look here. So first we'll talk about metabolism, and just know that there are different chemical reactions that take place within the cell. Either we're synthesizing, we're building something, or we're breaking down something. So know that your skeletal system is hard, right? Just like I'm handing on that table there, it's a hard structure. But unlike my table, it's a live structure. So the bone will be involved in remodeling, breaking down bone and building it back up, synthesizing it, creating new bone, okay? Replacing the old bone. So there are processes within the body that are involved in anabolism or anabolic processes and catabolic processes. Anabolic processes or anabolism, right? Process of building up, synthesizing. Think about anabolic steroids. You've probably heard that term before, right? So anabolic steroids or an anabolic process would be a process, right? You get big muscles and you get big and strong. Well, okay. Anabolism, process of building up, synthesizing. Catabolism, it's the opposite. So these are the opposite of each other, and it's the process of breaking down. Okay? ATP, I said to you before there regarding energy source available to the cell and to the cells of the body and the tissues of the body, very important, produced by the mitochondria, right? And really how we measure as far as the calories, we've all looked at on the back of different types of food that we eat and such, a caloric intake, right? Measure of energy contained within the food that we eat. Now, you'll see here as far as reproduction of a cell, okay? So all the cells of the body, except the sperm and the egg, right, for reproduction, all the other cells within the body are reproduced via mitosis. Meiosis is how these gametes, these sex cells, these sperm and the egg are reproduced, okay? So cellular duplication, mitosis. And cytokinesis, this is the actual division of the cytoplasm. And I'll show you where this takes place. So interphase, and I'm going to show you a graphic that'll be very helpful to you. Interphase is really where the cell life is primarily lived, right? This interphase. Mitosis is only a small portion of the cellular life cycle, and cytokinesis takes place during the latter portion of mitosis. So this image here, I'm going to repost this because I've added this to the PowerPoint, but I like this image. It's really very helpful to think about what's taking place in the cell life. So the cell cycle, the life cycle of the cell. Well, here we have this area right here is interphase. Primarily, the life of the cell takes place here. Cells mature, DNA is copied, cells preparing for division. Only a small percentage of the cell life is spent in mitosis. Now, you've probably heard this term mitosis, cell division. It's PMAT, prophase, metaphase, anaphase, telophase. Now, there's hand motions that I've learned, but I didn't create this, but I think it's pretty cool. Prophase, metaphase, anaphase, telophase. Now, at the end, telophase there, we have two identical daughter cells. So we start with a parent cell, right? And prophase, what happens is that there is genetic material, right, in the nucleus. Well, that genetic material then will be able to come out of the nucleus because the nuclear membrane will be dissolved, okay, in prophase. So prophase, nuclear membrane is dissolved. Genetic material in metaphase comes to the midline of the cell. In anaphase, then the centrioles and the protein structures will pull the genetic material to the separate parts of the cell. And then in telophase, you have two identical daughter cells. Now, see where it says cytokinesis here? At the end of the latter portion of anaphase and through telophase, the division of the cytoplasm, the guts of the cell, will take place at the end of anaphase and into telophase, so that we end up having, at the end of telophase, two identical daughter cells. Okay, that's what you're seeing here. So one cell takes place most of its life in interphase. Then division will take that one cell and create two identical daughter cells. And so you need to know, and like I said, this will be helpful as far as, I like this image. I think it's very helpful to you all. And what I'll do is I'll just post the image itself. How's that? Yeah, I'll just post the image in documents and resources. This way you have that specific image that you can look at and help you. And you're just seeing here as far as the different, what's taking place through the life cycle here. But what I'm going to show you, folks, is that, show you an image here online. So if you were just to look up mitotic division, and what you'll see there are all types of images that are present for you when you're doing the cell cycle. And you'll see here that, let's look at this one, okay? Here we go, okay, that's good. Okay, so what you're seeing here, folks, is that through the different cycles here, okay, we have prophase, metaphase, anaphase, telophase, mitosis. Interphase is where the primary life cycle is. So when we talk about mitosis, it's not interphase. Interphase is the life cycle, part of the life cycle, but mitosis is the division portion of the life cycle, okay? And you're seeing here, as I did the hand motions, right? So prophase, the genetic material is gonna come out of the nucleus. Metaphase, they all line up the genetic material right in the center of the cell. Anaphase, we're pulling them to different sides of the different portions of the cell. Telophase, we're actually creating then those two identical daughter cells. And cytokinesis is taking place to actually separate the cells into two identical daughter cells. So you'll see here, prophase, the chromosomes appear condensed and the nuclear envelope is not apparent, right? The nuclear envelope, the envelope that holds the genetic material together, it's dissolved. Metaphase, thick coiled chromosomes, each with two chromatids, that's what you're seeing here, these two lines, are lined up on the metaphase plate in the midline of the cell. Anaphase, the chromatids of each chromosome have separated and are moving toward the poles. They're moving to opposite ends of the cell. Telophase, the chromosomes are at the poles and are becoming more diffuse. The nuclear envelope is reforming. See, now you see that here, the envelope is reforming. And the cytoplasm may be dividing. That's that cytokinesis. And then at the end, we have two identical daughter cells at the end of mitosis. Now, meiosis is a matter of Okay, so in meiosis, what's taking place is that we have, it's like you have two divisions that are taking place, okay? Two divisions that are taking place, and we're going to actually start, we're going to create then 4 individual cells, okay, at the end. And this is only for gametes, for the sperm and the egg. And what will happen is that the sperm and the egg will only have 1/2 of the genetic material required for the cells of the body, right? Because when the sperm and the egg come together at fertilization, then we will have a complete set of chromosomes, a complete set of the DNA present within the cell. That's very important. So you'll see here that, here again, yeah. With mitosis, we end up having one parent cell, 23 pairs of chromosomes, right? So there's 23 pairs of chromosomes, 2 pair, right? So we have here then, at the end, we have 2 individual cells with full sets of chromosomes for each. In the case of meiosis, we're going to create cells that only have 1/2 of the genetic material required for a cell. So that again, like I said, when they come together at fertilization, they create a full complement of DNA. Okay, so I mentioned to you about the cytokinesis already. And so you'll see here reduction division, because we're reducing the amount of chromosomes present for these cells that are the sperm and the egg. And the gonads would be, so the gonads would be the ovaries for the female and the testes for the male. And 2 divisions resulting in 4 cells. And you're seeing here as far as that PMAT is present within meiosis, but it's stage 1 and stage 2. And at the end, we're going to form then those cells. So now here we have this term here, spermatogenesis, oogenesis, right? Now, 4 cells are produced as a result of spermatogenesis, right? This meiotic division, meiosis, and they'll develop into sperm. In oogenesis, it's a little bit different, folks, in that 4 cells are produced, but only one of those 4 will be a functional egg, ready to be released upon ovulation, and then the possibility of fertilization. Okay, very good.